I'm going to bring up Dr. Dan Chow. He's Vice President of Clinical Development at Surrozen.
Hi, everyone. I'd like to thank the organizers for allowing us to present at this innovation showcase. My name is Dan Chow, Head of Clinical Development at Surrozen and a retina specialist. We are a publicly traded biotechnology company focused on developing next-generation therapies for retinal diseases. These are our legal disclaimers. So first, a little introduction into Surrozen. So we are leaders in Wnt pathway modulation. We were founded almost a decade ago by the scientific leaders who discovered the pathway, and we've uncovered a lot of novel insights into how to translate this Wnt biology into therapeutics, now with a focus in ophthalmology. And we've done this by building a proprietary antibody platform, which is built on our foundational IP and how to modulate the Wnt signaling pathway. And that's led to development of multi-targeted antibodies, which we believe have best-in-disease potential, with our first development candidate having been licensed to a strategic partner. I'm really excited to tell you today about our lead candidate, SCN-8141. This is the first bifunctional antibody, which combines Wnt agonism, which you heard about in the past presentation, with VEGF inhibition, with the IND planned next year. Tony did a great job introducing the Wnt pathway, and we certainly see this as really a foundational mechanism for vascular repair, which until very recently was not druggable. And you can see here this Wnt pathway results in a number of processes very important for vascular health, such as maintaining blood-retina barrier, promoting vascular repair and reperfusion, as well as preventing neovascularization, which we think are all important properties which can enhance and also synergize with our anti-VEGF standard of care. The importance of this pathway has been well documented, both through human genetics, where mutations in these Wnt signaling proteins lead to human disease, characterized by incomplete development of the retinal vasculature, and then also with clinical validation, with clinical data with a Wnt agonist showing initial safety and biological signal in diabetic macular edema. Our approach, again, at Surrozen is to use these novel insights for our antibody platform to very robustly and specifically activate Wnt signaling. And now we've even gone beyond that and have evolved that platform to also incorporate additional functions where we can not only modulate Wnt signaling, but also other drivers of retinal disease. This is manifest in SCN-8141, which again, as I mentioned, is the first bifunctional Wnt antibody in ophthalmology. It combines dual mechanisms of Wnt activation and VEGF inhibition in a single molecule. And we hope that this can help to improve outcomes in patients with retinal vascular diseases. This is some of our preclinical data showing superiority to aflibercept in a well-characterized oxygen-induced retinopathy model. There are two key phenotypes of this model. On the right-hand side, first is the development of neovascular tufts, in which 8141 is able to very robustly reduce, superior to aflibercept. But then on the left, I think, which is even more remarkable, is also the ability to reduce the avascular area or to actually revascularize the retina. Aflibercept has no effect in this model, and it's well known that anti-VEGFs have not been able to show reperfusion of the retina. But remarkably, 8141 is able to almost completely revascularize the retina in this model. So we're really excited at Surrozen about the potential of combining these mechanisms of Wnt agonisms and VEGF inhibition, and excited to bring this into the clinic next year. Beyond that, we also have additional assets, all founded on a foundation of Wnt biology across other ophthalmic indications. Our next molecule, SCN-8143, is a trifunctional antibody, which combines Wnt agonism with VEGF and IL-6 inhibition. We have SCN-113, targeting different Wnt receptors, which has shown really promising preclinical data in Fuchs endothelial corneal dystrophy, as well as models of GA, as well as our original candidate, SCN-413, being developed for retinal vascular diseases in partnership with Boehringer Ingelheim. And with that, I'd like to thank you for your attention, and please reach out for any questions.